Hi, my name is Maria Deutschmann, and today I invite you to discuss microbial network change in the global ocean. Before that, let me introduce to you the microbial networks. Microorganisms are very tiny, and there are a lot of microorganisms, especially in the ocean. They are also very important because they contribute to the ecosystem functioning. Lastly, microorganisms are the basis of food webs. It's not only important to know who is there and in which quantity, we also have to know who is interacting with whom. However, microbial interactions still remain barely known. One of the key problems is that most of the microorganisms cannot be cultivated. And even if we could cultivate all of them, there are just way too many to experimentally test all potential interactions. In order to identify and quantify microorganisms, we use their genetic material. Usually, we use the 16S and 18S rRNA genes in order to measure microbial sequence abundances. These marker genes are very handy because they are conserved enough to compare and diverse enough to identify microorganisms. When we measure microbial sequence abundances at the same location across time, we have temporal data. And if we measure it in different places, we have spatial data. Both types of data can be used to study microbial interactions via association studies. I envision the microbial associations as a network. A node is a microorganism, and an edge represents an association between two microorganisms. The network approach has been employed already in the past years, and it gains more and more popularity. Networks are a very handy tool because they can handle the high number of microorganisms and their potential interactions. So they allow us to explore potential microbial interactions, and we can use them to elucidate patterns of the microbial ecosystems. Still, microbial interactions remain barely known because there are challenges. First of all, the network is not looking as pretty as I showed you previously. It's rather looking like a hairball, so they are very dense with most nodes connected to the rest. One key challenge is that an association may or may not represent a true interaction, because after all, correlation is just not the same as causation. Another challenge is that usually we use a static network to investigate the microbial ecosystem, which is rather dynamic. So it changes in time and space. Does the network also change in space? This is the context of the project I present to you in my poster. And the key aim is to disentangle the spatial distribution of microbial associations in the global ocean. We have data from the major oceans, the Mediterranean Sea, from the surface down to the deep ocean. In the poster, you will see that I employ a network-based exploration via sample-specific subnetworks. I first construct a single static network, and then I determine 397 sample-specific subnetworks. I use them to compute a spatial recurrence score for each single microbial association. Please come and visit me virtually in my poster if you're interested in how the network changes across space, if associations are everywhere, and to discuss with me the hypothesis if a similar network leads to a similar network structure. I'm very looking forward to talk to you how a network can be used to study marine microbial associations in the global ocean. And with that, I would like to thank you for your attention and also thank my collaborators, especially the PhD supervisor Ramiro Lugares. If you would like to see a network popping up in your Twitter now and then, don't hesitate to follow me. Also, thank you to the NetBio Cozy organizers for letting me present my work to you.